You're listening to Exposed by the So Cultured podcast.
We're your hosts, Liv,
Yaz,
and Taz.
Three girls with a passion for science and tech.
Yep, and welcome to our thrilling new series.
This series, we are taking you on a journey to uncover the truth behind your fave TV shows and movies.
From the intergalactic adventures of Star Wars to the monstrous depths of the Meg.
And the chilling scenarios of zombie apocalypses.
Join us as we question whether these plots could ever happen in real life.
And reveal the scientific inaccuracies hidden on our screens.
And don't worry, we will still be dishing out the LaboraTEA this season with some even crazier listener stories than ever before.
So grab your popcorn for a side of piping hot LaboraTEA.
Before we get into it, remember that all opinions are our own and all our listener stories are alleged.
And now let's get into this week's episode of Exposed.
Hey everyone, and welcome to the season finale of Exposed by the So Cultured podcast. I cannot believe we are at the end of this season. Where has time gone? We are genuinely so upset to see this season come to a close because I feel like this has been the best season so far. But I feel like we say that every season. We're like, oh, this is the best season. I definitely think it is just so nice to learn about films. Like, I would consider myself a movie buff. Like, I love films. And this has just like exposed so much that now I'm just like, damn, I don't trust directors. Or I want there to be a job. Like, I want to be the science, like the science consultant.
Scientific
Is that like
advisor.
a job?
Yeah.
Advisor,
I told
do
you,
you know what I
remember,
mean?
I told you last week in the Interstellar episode that there's the guy, Kip Thorne, and he's like a theoretical physicist, and he was actually hired to like advise them for this film and like give them all of the, you know, the scientific details. Like,
I'm doing that. That's going to be a new job role for me.
Mhm.
But it is so nice to learn the science and the tech of like facts or fiction in such a fun and refreshing way. I'm just obsessed.
Yeah, I agree. And like, as we said at the start of the season, that's something that we really want to achieve with this podcast. Like, when us three first started talking about this podcast, we always said like we want it to be fun, we want it to be different to all of the other podcasts, like science podcasts that exist. So we want every season to feel like new and refreshing for you guys, and yeah, like Yaz says, like help you learn in like fun and different ways.
Yeah, exactly. And with that being said, best believe we do have another exciting theme lined up for season 4. I mean, I can't even believe that we're already talking about season 4. Like, what the hell? It seems like yesterday.
Honestly, it feels like yesterday that we started this podcast. I'm so confused that we are going to be doing season 4. Actually makes me feel ill. Like, we've been going ham, like, proud of us.
We have.
But also, it's been like a year. Like, we started it in January, didn't we? Or it was just after Christmas. We were like, oh, let's actually make it a reality. So time has definitely flown by. And I'm just so glad that we've actually got so many people who tune in every single week. Like, I don't think I would have expected that at all.
No.
And we genuinely appreciate you guys so much. So thank you for following along, staying interested and engaged in our podcast. And without you guys, this podcast would not be anywhere near as fun. So we appreciate all of your interaction, your engagement, your comments, messages. We love you all because literally your messages help us keep going, because it's just nice that you guys enjoy what we're doing. And we also enjoy bringing this to you as well. And we just hope that we continue to bring you a podcast that you feel excited to tune into every single week.
Yeah, that is so true. And we do love you guys.
Yep. And with all of that being said, Liv, you always give us a question of the week, and I'm obsessed with it. So go on. What is the last question that you have for us this season?
Okay, so yeah, my question of the week, the last question of the season. So we are all scientists. We are all very fact-based or like evidence-based people. And often I feel like what comes with that is that I think I am not a very creative person. So I've been trying to think lately of different ways to like get into my creative era, like different things that I can do to be and feel more creative, because I feel like that's something that I miss a lot in like my personality and also my life. So I was thinking about like, if I was to design something or like work on some kind of project where I'm doing some like designing or something creative, what would I do? So my question to you guys is, if you had to design something, it can be anything, what would you guys design? Because I need some inspo. So like, I've got an idea in my head and it's definitely not something that I'm going to execute, but I would love to design like poleware, because I can never find poleware that I feel like really like speaks to me and the kind of like design choices that I usually go for. So I would love to like get into that, and I see a lot of people in like the pole dancing space getting into like designing poleware, knee pads, loads of different random stuff, and I would love to do that, but like it's definitely not something I'm actually going to pursue because I just don't have the time. But like that could be like a good example, like maybe you wanted to start designing something, I don't know, that would come in useful to you.
Yeah, maybe cute lab coats.
Yeah, that would be
Or like
a good one.
embroidery on lab coats that are like sciencey, or you know how like people get patches, or even like the transfers that you could like iron on, you know, and they like stick, so it's like embroidery. We could do that for like lab coats, but really cool science themed stuff. Maybe there's already that exists.
Well, there you go. Yeah, if you could make some transfers, then people could personalize their work ones. So cute idea.
Yeah, but also our work ones aren't like assigned to us. Because they get washed, then you just like pull one out of the like clean kit. So it's not like you have.
So
a
who do you
specific
think is
one.
going to use your transfers and your embroidery then, Yaz?
Yeah, I was gonna say.
Sorry, that was a bit savage.
Well, I'm just being
Flawed
creative
idea
here.
from
You
the
asked
off.
Flawed
Try
idea.
again.
asked for creative input, and that is my creative input. It's relevant to me, I think. Right, listeners, write in, comment, message us if you would buy my embroidery.
You can make So Cultured ones.
Well, I was gonna say that I would design So Cultured merch because
Oh, perfect. Do you know what, Yaz? That is a perfect idea. Perfect answer.
yeah.
Do you know what as well? Like so many of our listeners actually, and Taz is actually wearing hers right now, but so many of our listeners ask us about the hoodies, and best believe, guys, we will one day, hopefully soon, have them available so that you guys can like wear them, because you ask us all the time how you can get your hands on one. So maybe Taz, there you go. That's what we need to do. And I feel like that's something I could actually implement in my everyday life, and it'd be a really good way for me to like get more creative and like design things. So
Yeah, like stickers and things like that.
yeah, love that idea.
Yeah. All right, let's get on with today's episode, because we are actually going to be talking about designer babies and the film Gattaca. So let's get into it. Okay, guys, so for our final episode, we have a film that I actually hadn't heard of before, but it was requested by a lot of our listeners, so we wanted to include it. And it's actually known as being a hard science fiction film, which is actually a sci-fi that tries hard to be as scientifically accurate and as logical as possible. So from what I could see online, a lot of people were actually given this assignment by their biology teachers, which is quite cool, especially during quarantine. That's what Reddit was saying anyway. So yeah.
Yeah, until now, I'd also not actually heard of it. So do you want to tell us and the listeners what it's all about?
Yeah, Gattaca is a science fiction film, and it was released in 1997 and directed by Andrew Niccol. So just straight off the bat, the title uses the 4 nucleotide bases found in DNA, with G relating to guanine, A to adenine, T to thymine, and C to cytosine. And it's pretty appropriate because the entire film is kind of based around a future society where gene editing or genetic engineering has become commonplace, and it allows parents to select desirable traits for their children to ensure that they're born without genetic defects or like predispositions to disease.
I'm not gonna lie, I don't know why I've not heard of it or seen it, because this sounds so cool.
Yeah.
Like what? And also it was made in 1997, so I was literally like 2 years old, so that seems like so ahead of its time, because CRISPR wasn't even a thing. Like that was only a recent discovery. So like what the hell, like I do think it's mad when films are so futuristic and you're like, how did you even think of that? Like, that's so ahead of its time.
Yeah.
So what actually happens in the film? Because I guess, like, if we've not seen it, probably a lot of our listeners also have never even heard of it or seen it.
Okay, so in the world of Gattaca, the society is divided between genetically engineered vitros or valids. So like in vitro fertilization, it kind of like plays on that kind of word, who were perceived as superior due to their enhanced genetic makeup. So like the selected genes, and uteros or invalids, who are born naturally and obviously carry the genetic imperfections, which is then deemed undesirable by society.
Invalids.
That's
Yeah,
so sad, isn't it? Like,
invalids.
you're an invalid.
Yeah, but that's like invalid, no? Isn't that such an offensive word? You can't say that.
Yeah.
Well, it kind of plays on that, yeah. And there was another, they said also in the film, instead of saying he was a degenerate, he was a degenerate. And I was like, that is so clever. I love it.
That is clever. I like the play on words. Like, I like this.
Yeah, you guys should definitely watch it because it's actually really good. But anyway, the story follows Vincent Freeman, an invalid who dreams of becoming an astronaut despite his genetic shortcomings. So Vincent Freeman is introduced as a child born without genetic modification, faces discrimination and limited opportunities due to his genetic inferiority. So even when he's born, they supposedly determine his life expectancy, which was around 30 years old, and then also the likely cause of his death, heart failure. So like from then on, he was kind of discriminated against because they had this information about him. And even in the film, they show that he can't attend school because their insurance in the school wouldn't be able to cover him. Like, he would be too expensive for them to have at the school. And then a key point in this film is that due to this discrimination that he faced, his parents then decided that for their second child, they would get a gene edited or a valid child. So first they decided on the gender, and they decided a boy because they wanted Vincent to have a brother. And then they went on to edit the appearance of their child to also kind of look like his brother and like what they wanted. And then they removed disease traits, and then even at this lab where they were at, the scientists said, oh, I've gone ahead and removed undesirable traits like male pattern baldness, myopia, which is short-sightedness, alcohol dependency, susceptibility, and also predispositions to obesity. And then the film then goes on to show that compared to his brother, Vincent does inherently have less advantageous genetic traits, but that paired with the belief system that invalids are of lower caliber and class deems Vincent to be like undeniably disadvantaged in this like new society.
Aww.
Yeah, like just because he had to wear glasses in the film was just like a sure sign that he was an invalid. So that was like one of the main things that they played on. Yeah, me too.
We would not be accepted in this society.
Oh, I feel bad for him. Like, I'm actually invested in him and his life. I do actually, it sounds really interesting actually. Like, I think I'm going to give it a watch, to be honest. But like, when you mentioned about him wanting to be an astronaut, I was like, oh my God, that's so cute. Did he get to become an astronaut? Because you said he can't go to school, and like, how is he going to become an astronaut if he can't go to school? Did he get to become an astronaut or no?
Exactly. Well, Vincent himself becomes like super determined to achieve his dream, but due to the prejudice, he kind of needs to bypass this low social status somehow. So Vincent actually meets this guy called Jerome Eugene Morrow, a valid who was genetically engineered but became paralyzed in an accident. So Jerome actually agrees to help Vincent by providing his genetic material, so like his blood, his hair, skin cells, and urine, so that Vincent can assume Jerome's identity and pass the genetic screening tests at Gattaca. So they showed that his whole interview was just a urine sample, and they tested that just to make sure that he was a valid, and that was it. That was the only entry requirement, because in their mind, he's so genetically edited credited that he is like perfect for the role. Like you don't need any
my
other thing to
gosh.
determine it.
Mm.
So basically the whole film centers around how he faces numerous challenges and close calls as he tries to maintain his disguise. So I won't go into the rest of the film just in case you want to watch it, but that is like the crux of it, really. But something basically threatens his identity to be exposed once he qualifies for a space mission that he's been dreaming of. So then the film kind of goes into that. But essentially the main themes are like the ethical implications of genetic engineering and the societal consequences. And I guess how genetic makeup alone doesn't always determine a person's capabilities.
Yeah, damn, this is a cool concept. Like, I
Yeah.
am gonna get off of this call and I'm gonna watch it. I hope it's available on like Netflix or something. So I guess
Yeah.
the main thing we are going to expose in today's episode is going to be all about like genetic modification and designer babies. Is there anything else that we're also going to cover?
Yeah, exactly that. So that will be our main focus for the episode. But before we get into that, I want to talk about the dystopian future that they're in and how accurate or not so accurate it was. But firstly, the thing that struck out to me so much was their electric cars. So I watched the film and I thought it was quite wild how right they got electric cars, because they had Tesla-like charging stations outside their house. And I just thought, like, 1997, I really hit that nail on the head for a film made 27 years ago.
Oh my god, 27, stop, because I was born in 1997, so like that makes me feel really old, because like you're like, oh yeah, it was 27 years ago, like
Just exposed your age, sorry. So that was one thing that really struck out to me, that was like so impressive that they got that right. Obviously that, and the whole like genetic modification aspect to it as well. But what doesn't quite line up is the regular human space travel to and beyond Jupiter. So this idea is still completely removed from our current technological capabilities, and the notion of human travel to Jupiter now seems even further in the future than it did then. Because if you think about it, they were, well, it wasn't 1997 that they went to the moon, but like obviously it was more fresh in their minds, like traveling to space and stuff, than it is now. Because like I think December 2022 marked half a century since humankind last stepped on the moon. So it will probably be a few more decades until astronauts are traveling Mars or orbiting Jupiter. So yeah.
Yeah, and that's like a whole conversation in itself of like, why have we not been back?
Yeah.
But I do agree with you, like back then they probably felt more positive about space travel than we do now, because I think we've discovered so much since then, and we've spoke about this on previous episodes of this season actually, but like we've discovered so much then that probably now makes us think that it is actually even more out of the realms of possibility. So yeah, that's a really interesting point. But let's get into like genetics and designer babies. Was it an accurate representation? How do you feel about the science like represented in the films with regards to that?
Yeah, not only was the science pretty accurate, but the ethics and the ideas surrounding it too. So Gattaca was actually released about a year after the first mammal, Dolly the sheep, was cloned, and actually only a couple years before the June 2000 announcement of the first working draft of the human genome sequence.
Oh my god, they didn't even have the human genome sequenced.
Exactly. So it was very, really like early days, but that also meant it was a time where there was real ethical conversations around, I guess, gene editing and cloning, and that's where it all started happening. So since the movie came out, science has obviously produced rapid, accurate, and cheap genome sequencing, genome-wide association studies, and precision genetic manipulation tools such as synthetic biology and CRISPR. And Gattaca essentially universalizes the fears and the potential dangers of this exact science if it was unregulated. So again, you can kind of see how it was quite ahead of its time and kind of really hit the nail on the head as to like maybe some of the conversations that are happening today. But what can we actually do today? Well, first up, genetic testing of prospective parents is pretty commonplace, especially if one or both parents have familial or genetic predispositions to certain diseases. So for example, in my family, we actually have a type of blindness that that runs on my dad's side of the family. So that is something that maybe I might test for in the future. I think it actually runs on the male side, and my dad doesn't have it, but my 2 uncles are both blind on my dad's side. So I think my cousins may have to consider genetic testing if they want children, but I think it probably ended with my dad, so I'm not sure I have to, but I would have to check that anyway. But obviously something like that, and also more commonly cystic fibrosis or Huntington's disease, where passing on the genes encoding those diseases would be detrimental to your offspring, are commonly tested for in parents. And not only that, but couples at risk of having a child with severe or life-limiting diseases such as cystic fibrosis or diseases that might cause muscular dystrophy, they can actually use pre-implantation genetic testing to actually select among embryos created through in vitro fertilization or IVF for those that don't carry the disease causing gene. So they will test the embryos that they have, and they will only implant the embryos that don't have the disease causing gene, because obviously it's not always passed down. This is where it gets a bit dystopian, right? Because countries like China, Brazil, Japan and Singapore allow for sex selection for non-medical purposes, but with certain restrictions. Then the US and Mexico also allow non-medical sex selection without restrictions. So in those scenarios where you're already doing IVF and you want to do genetic testing and you find out 3 of the embryos are female and 3 of them are male, you could decide I want a male baby, which I think is wild that you're allowed to do that.
I thought that was illegal.
No, apparently it's not. Not in certain countries. It's illegal in the UK, but yeah.
What?
Yeah.
That is mental. But I guess it's because the majority of people aren't using IVF. I think it would really become a problem if rates of IVF became really high and then everyone is doing non-medical sex selection or whatever. But for now it's probably not, is it that big? I mean, it is a big deal, but it's not
yeah,
we're gonna completely eradicate.
Not big enough to have an impact. But I just think it's so crazy that if you have the money, then you can.
That is mental.
I think it is quite bad though, because in certain countries, I'll assume with China, if you can select to have a male child, then you're going to carry on the name. A lot of the problems when there was the one child policy, people would literally dispose of female babies because they were we don't want a girl, we want a boy. And then there was so many issues. So I feel that's so problematic that they'd literally just be selecting for men.
Yeah, so when I was doing the research for this, they actually did put a ban in place for sex selection, but I think it was only 3 years long. But I think there is more restrictions around it in China than other places in the US, but I don't obviously know all the ins and outs on the actual regulations and restrictions. So, but it says non-medical restrictions, so yeah,
that's mad.
But obviously we're talking all about designer babies and stuff like that today. There's a number of organizations that actually take this whole aspect to the next level, in my opinion. One company which I was researching called Orchid do genetic testing on parents prior to conception to actually calculate risk scores, estimating their own likelihood of confronting common illnesses like heart disease, diabetes, schizophrenia, and then the likelihood that they will pass those risks along to a future child. So parents-to-be can then use IVF along with an embryo screening package to identify the healthiest of their embryos for pregnancy. And this type of testing goes a step further than just testing for specific diseases, but more common diseases to identify, among various embryos, the future children least likely to develop things like heart disease, breast cancer, prostate cancer, diabetes, and other conditions. And that is referred to as embryo prioritization.
Yeah, I have actually heard about all of this because, this specific part that you just said, because I went to a Pint of Science talk once where one of the professors at my university, he studies this and he was going to talk about it. And it was really, really interesting, even though it is it sounds really crazy, but I actually can see the benefits of it. And this is like a bit random, but I actually remember once I was watching on YouTube this documentary about an incest couple. So it was a father and a daughter who were in a relationship.
Ew.
And they really, they really wanted to have a child. And they were actually talking about like how they were like going to get genetic tests to like see if their baby would be healthy, because obviously like, you know, there's a lot of problems with incest breeding,
Yeah.
right?
Yeah.
Breeding, that sounds a bit weird, but yeah. And I heard about it all there, but I am a bit like skeptical on something you said. So you mentioned like generalized diseases, things like heart disease, breast cancer, and all of these. But we know that these diseases are polygenic, right? So they can be caused by like several different things going wrong in different genes and different parts of in your DNA. So like, how are they estimating it? Like, I don't get it.
Well, that is exactly the problem with this whole idea that they can select for something as general as heart disease, because that runs in families, just like height or your musical ability. And only really in exceptional cases can the inherited risk be traced back to a single gene. So that is what Liv meant by polygenic. Like, it's caused by hundreds and thousands of genes contribute in a small way to something like heart disease. So that's what it means to be polygenic. So it would be hard to use these polygenic risk scores as a way of selecting embryos, because essentially we don't know what different variants are doing what biologically. And a gene variant that may be associated with a decreased risk of breast cancer could be associated with an increase of your risk to something else. So not really enough is known to be able to do this, in my opinion.
Yeah, honestly, that is actually wild. I do think that, well, I actually never knew that you could do like non-medical sex selection. Like, I actually had no idea that that's a thing that you could even do, or even like in the way that you can kind of genetically select for certain things with babies. Like, do you think that's playing a little bit on like the evolutionary aspect? But I guess if we're talking about like gene editing, I remember in the news, wasn't there some like twins or like babies in China that they were like genetically editing? What's the case of that? How does that work?
Yeah, so basically when CRISPR first hit the scene, the idea of designer babies was actually brought up again, because like you said, Yaz, so far we've only selected for desirable genes, not edited the actual embryos. So now we're talking about removing troublesome DNA and replacing it with something else. And not many would argue against altering a gene that might cause a deadly disease, but why stop there? Why not change the eye color or make your child smarter? And that was for a while an academic question. And there was almost like an understanding among scientists that they could experiment even on human embryos, but they wouldn't implant them or bring them to term. And that all changed in November of 2018 when the world actually found out that He Jiankui I'm so sorry if I butchered that pronunciation, a biophysics researcher, and he created the world's first genetically engineered babies. According to he, the two children known as Lulu and Nana had their CCR5 gene modified. That gene is actually responsible for a protein that allows HIV to enter the cells, and modifying it, if successful, could mimic a mutation found in about 10% of Europeans, which actually protects them from the virus. So it's modifying this protein that HIV usually uses to enter like white blood cells. So by editing this particular gene that encodes that protein means that HIV can't enter. And actually there is a percentage of the population that have this mutation. So they're like,
I've never heard that before.
Yeah, so they're immune to HIV. I think the reason they found this is because there was someone had HIV and they also got a type of blood cancer, which meant that they had to have a bone marrow stem cell transplant. And they actually got a stem cell transplant from someone who had the mutation. So not only did they cure the blood cancer they had
and the HIV,
they cured the HIV because he was then immune to HIV, and that's how they discovered this mutation. And
so could people that have this, obviously it sounds a bit like, just get them in a lab and just keep doing blood transfusion, or like keep taking their blood, but couldn't you just give their blood to people that have HIV?
So it would have to be a bone marrow transplant, because that is
Okay.
where your like blood cells are made. So if you like obviously removed their own bone marrow and then replaced it with
And
someone who
okay,
had
so it's
this
quite invasive.
mutation. Yeah. So that's why it's not become commonplace. But like, yeah, if someone gets a blood cancer and they also have HIV and they had to have a bone marrow transplant, then it would be then beneficial to take the bone marrow
my
transplant from
gosh.
someone who had this mutation. So yeah. Okay, so going back to Hay and his activities, part of the concern about his activities centers on the fact that he performed germline editing as opposed to somatic editing, which is commonly used on human patients. So when we talk about gene editing, it's usually done on our cells that isn't going to be passed on to like our children, right? So any gene editing is just done on our like normal cells, not our germline. But Hay actually performed this editing on a germline. And so by editing embryos from the jump, he created a germline change that could be passed down to any future generations. So essentially, he didn't just change the DNA of two individuals, he potentially changed the entire gene pool from that point onwards. So consequently, Hay was banned from performing science and sentenced to 3 years in jail, which I don't think is that much, to be honest.
That's not long.
No.
Yeah,
Wait,
exactly.
did he do all of this in secret? Like nobody knew he was doing it?
Yeah, he did it all in well, there's conspiracies around that, in that the Chinese government were kind of like not really opposed to it because,
Right, okay.
but that's like a big conspiracy. But yeah,
Yeah.
he was doing it in secret.
Mm-hmm.
And also that's probably another reason why he only got 3 years in jail, because the government weren't really
They weren't really
necessarily
mad.
opposed to it. It was
Yeah.
just like on the global scale, he got really
Yeah.
criticized.
Yeah.
But yeah, that's because Hay skirted safety protocols and obviously conducted illegal medical practices. And what's more, we don't know what this effect would have had on the babies. So CRISPR isn't always as precise as we think it is, and it can cut unintended parts of the genome or may be repaired incorrectly. And additionally, in its normal state, the CCR5 gene actually protects against other diseases, and changing it could then make the children susceptible to infection down the road. And then on top of that, there has been some additional speculation that the children may have inadvertently have had their brains enhanced, because in animal studies, knocking out the CCR5 gene actually resulted in improved memory and cognition performance. So
That's fucking mad.
it's worth noting that it's unclear if the same result occurs in humans, but obviously time will tell.
Did the baby survive?
Yeah, and it
So
was
they're like,
2 babies.
Were they twins?
Yeah.
Oh my god, imagine. I mean, if that whole like improved cognition thing is real, that's taken like privilege to a whole new level. Like,
Yeah, I mean,
insane.
it's literally
Isn't
exactly
it, though?
like the film,
Like,
right?
yeah.
That's how
That is,
scary
yeah.
accurate it is.
Oh my god, yeah. I forgot for a minute that we were talking about the film. Sorry. I just forgot for a minute because this story was so interesting. But that's actually true. You are right. That is literally like the plot of the film seems very like outlandish and like not ever gonna happen. But like technically it's actually
It's
that's
actually
mad.
happened.
You can see why like watching this film, like a school would like set it as like a biology assignment.
Yeah, and given that advancements in genetics like continue and perhaps even supersede the innovations of the film, Gattaca is relevant even 27 years later when discussing like the ethical, legal, and social implications of genomics and bioengineering for scientists. And it therefore remains as a feature of the genetics and bioethics syllabus in academia, which I think is so cool, just because it's so relevant. Like there was even a Nature paper on Gattaca, the film, and how relevant it still is, and like the ethical considerations.
That's so cool. Do you know what? Like, and I think a lot of the films we've covered, there's actually scientific papers on these films. So like I think that's really interesting as well. Like, oh my god, guys, maybe we should take all of our knowledge and go write some papers now.
Yeah, let's write a nature paper.
I'm not gonna lie, Taz, I think this
insane.
was my favorite episode because one, I've never seen it. You have sold this film to me that I
100%.
literally want to watch it. Some of the other films that obviously I've seen them, or even then I feel like, yeah, they're cool. Whereas this is like, or obviously for me, I do CRISPR in the lab anyway.
Yeah.
But oh my gosh, I literally can't wait to watch this. I hope it's good. A good
No, it's
film.
actually so
Well,
good.
Wait, I just want to say that like when I was talking about Jason Statham in the Meg episode, you
Oh,
said,
yeah.
"Oh, I'll talk about." So, what is there? We got a deal in this film as well?
Well, I mean, first of all, you've got Jude Law, who is an absolute spice in
love
this
Jude.
film. Then you've got Ethan Hawke, and then you've got Uma Thurman, who is also
Oh, I
a
don't know
spice
who he is.
herself. So, like, three
Wow.
So all-star
leading
cast.
roles, and they're all spicy, to be
Wow.
honest.
So we've got some pretty cool science and some good-looking people. I think you're in for
Yeah.
a good time, Yaz.
There's actually so many things that I didn't really touch on as well. Like basically in the film, to like get into the academy, he has to do like a finger prick to like enter, and there's this whole thing that happens that threatens his identity to be exposed that happens at the academy, and it's all about how he tries to prevent his DNA from going anywhere and how he plants Jerome's DNA in his place, and it's just so good. Like there's so many things that's so good, and the whole like gene editing of the embryos, there's a picture of the embryos in the background when his parents are selecting his brother, and it's all just very good. So if you're listening and you haven't watched it, you should watch it. That's really good.
Okay, yeah, we will definitely watch it. So yeah, thanks for that, Taz. Really good episode to end the season on. We will of course pop that nature paper in the episode description along with any of the other relevant science. And so with that really concludes the science of season 3, which I'm very sad about, I'm not gonna lie.
Yeah, me too.
But to finish off today's episode and to really round off the season, we have a very exciting stand-in for our LaboraTEA section today. So let's get into it.
Yay!
So as you guys know, in the final episode of each season, we like to do something a little bit different for our LaboraTEA section. And this is because we don't want to bore you doing the same old thing. If you heard last season, we did our big fat So Cultured quiz. So for this episode, we're going to do something completely different. And I feel like me, Taz, and Liv are actually so close. We talk about our lives all the time, but you know when you're like, oh, I've got such a close friend, but I actually don't know what they do for work?
Yeah.
Can I just say, like, have you seen those TikToks where it's like a group of girls, they get together and they do like a PowerPoint night, and it's like what they do for work, and they literally do a PowerPoint of like, this is what I do for work.
Yeah,
that is it.
I feel like I actually don't know what you do. Like, I'm kind of like, if I introduce you to my other friends, I'll be like, yeah, so she does like epigenetics, but like don't know what she works on. So what we're going to do is we're going to tell you guys and each other what our PhD titles are. And then you girls have to interpret it and like we have to interpret each other's and basically come up with a whole thesis strategy of like what you've done, how you go about doing your experiments or your coding, like what you actually do to answer the main question. I actually think like I actually don't know what your titles are. So who wants to go first?
Okay, I feel like let's do Taz, me, Yaz, because it will be like lab, coding, lab.
Okay. Cool. I'm actually nervous.
I'm feeling pretty confident. Like, I'm actually feeling confident about Taz. I'm going to smash this.
All right.
Really?
Okay, smash it.
Yeah, I'm feeling confident.
Right, so I feel like I'm graduating in graduation when they read out your whole thesis title and everyone in the crowd is like,
no,
oh my god,
they did not do that at my graduation.
Oh, they did it at mine. Only for like people who did PhDs. They like, and awarding for a PhD of this goes to Rebecca Shipley for the title of blah 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 blah.
Wow,
so I feel like
cool.
I'm
go on, do
again.
it in that like professional way, as if you're awarding yourself your PhD.
Okay. And the award of PhD goes to Rebecca Shipley for the title of Defining the antigenic requirements for pan-lyssavirus neutralization.
What's a pan-lyssavirus?
Sorry. Say that again. Say it again.
No, because what I know you work on was bats and rabies, and that's not in that title. So what?
Exactly. Start again, please.
Defining the antigenic requirements for pan-lyssavirus neutralization.
Right. Our first
Go on,
task,
Liv.
Yaz.
You were so confident. Give it to me.
Listen, we're a team on this podcast. We work together. Don't be singling me out.
I'm gonna smash it. Is it not a
Would you-
competition of like who's
Would you
the closest?
Would you like to smash it with me, or would you like to be left behind?
Oh,
I mean, Liv, you
fighting
didn't hear how
thought.
confident I was. I reckon I could, antigenic pan.
I'm gonna sit back.
lyssavirus
Yes, go for it.
So that my eyes are watering. So a lyssavirus, I feel like Taz has once explained that.
Yeah, I have.
Didn't she say pan-lyssavirus?
Yeah, so we're going to come to the pan in the future because that might be like another thing. I don't know. A lyssavirus, isn't it like COVID?
No.
COVID had a lyssavirus or COVID. My eyes are actually watering. What is a pan-lyssavirus? Okay, Liv, we're doing it together because I can't.
Yes,
I actually
thank
don't
you,
know these words.
Thank you. Let's work together. So pan-lyssavirus, like I want to say it's encompassing something to do with like bats.
No, but a lyssavirus, isn't that like a type of virus? It doesn't mean bats.
Yeah.
That's not another word for like bats.
But then the pan part,
What you get pan
What
and
does
bats.
what does pan generally mean? Like
frying pan, like
Pan.
you fry. I don't know. You put stuff in it.
Pan, is it like together or something?
Kind
I'm
of.
thinking Pangea.
Yeah,
Or like
kind
pangenic. Is that a word? Pangenic.
no,
I'm making
like
stuff up.
it's like all encompassing kind of.
All encompassing, yeah.
Pan-lyssavirus means kind of like all encompassing.
Right. All of the different type of lyssaviruses. Antigenic.
Yes.
What does antigenic actually mean? So what was it? It was something defining.
Defining the antigenic requirements for pan-lyssaviruses.
Neutralization.
Yeah.
Neutralization.
Right. Neutralize it. So you need to figure out how to combat all the different types of lyssaviruses to neutralize the effects.
Mm-hmm.
However, we're going to go deeper because we know that Taz worked on rabies.
Mm-hmm.
So rabies is obviously a type of lyssavirus.
Mm-hmm.
Yeah.
But what's the pan is just like, is there multiple lyssaviruses?
There's
in-
multiple.
Yes.
So she's looking at all the lyssaviruses. She's not focusing on one specific.
Why in bats?
Lyssavirus.
Okay, no, like we know why in bats because like we can like make that in like a grant proposal. You're like, bats is so important. There's bats everywhere. Like we need them for like fertilization. Blah blah blah.
Well, and
But they
bats
can cause
carry
disease.
bats.
Yeah.
Bats are animals that can carry all of the lyssaviruses.
Yeah, they're the
Right.
host reservoir species, which means like Ebola and COVID or like SARS. They're like thought to be the reservoir species.
So they were the perfect study organism for your thesis title.
Yeah, I didn't really study them. I just like that is just like the basis of where they come from and where they're what
Okay.
species they're detected in.
So lyssaviruses can get into humans as well, but you
Yeah.
want to figure out how you can like stop the effects.
Yeah, and how do you tend to do that with viruses?
Vaccines.
Yeah,
So the neutralization part is you were making vaccines.
pretty much, yeah. So I was
That's
defining
mad.
so
Damn,
my
not clever.
whole thing was defining what would need to be included in a
done.
vaccine to
Taz,
be
stop.
cross-protect-
Let us
Oh,
get this. I was going to say
okay.
that. I am clever. I
Sorry.
swear, like I was going to say that. Wait, can we go back to like what does the antigenic mean? Antigenic.
Yeah.
Let's break it down. Antigen.
Mm-hmm.
So it's like an antigen. And
Oh, yeah. So defining the antigenic requirements. Yeah, that does make sense.
let's break it down for any listener who's not scientific. What is an antigen?
An antigen is something that binds to an antibody.
Or the other way
to
around, yeah.
To neutralize the effects of a virus. No?
Yeah.
And your body produces antigens.
Well, no, the other way around. Your body produces antibodies to bind to
yeah, and then
antigens.
okay,
So
yeah.
did you do like ELISAs and stuff like to get the binding
Finding
of
the
antigens, or is it
antigenic
not that?
requirement.
I did that once
Yeah,
in my
I did do
undergrad.
ELISAs, but I
Oh,
worked
I got it.
with
It's like defining the antigenic requirements because the pan-lyssaviruses have antigens,
yeah,
and you need to figure out what needs to bind to the
Antibodies.
antigens to neutralize
Oh my god, we're there.
very
the virus.
close. Yeah.
What do you mean very close? That's
Wow.
it. We hit the nail on the head after
Innit?
like
What?
Like,
Okay,
what
so
else do you want?
what are vaccines composed of?
the dead virus.
Yeah.
Like not non-living, but it's not living anyway, so that's not actually correct. A weakened version of the antigen.
But like essentially how do vaccines work?
I actually think I know this.
Yeah.
Go on.
So what?
Wait, no, I was waiting for Liv to be like, I know Taz's
Nah,
project so well.
take the floor, Yaz. Take the floor. We're working together, babes. We're a team.
Okay, so teamwork makes the dream work. Vaccines work because you introduce like a weakened version or something of the virus, and then your body starts to produce the antibodies against the virus naturally
Mhm.
because of how they work with their white blood cells or stuff. And then like your body has it, like, because haven't you got like a primary and a secondary
Then your body
immune
has
system?
Yeah.
Like, no, you've got like the different levels of an immune response, and
Yeah.
so then if you actually get the
it.
actual
It makes
virus,
memory T cells,
memory
B cells.
cells.
Yeah.
B cells, memory B cells.
Yeah.
and then when you actually are infected with the virus, your body can just be like, bro, we know what this is before, let's just like make them again,
Yeah.
the antibodies.
So your body makes antibodies, and the antibodies directly target antigens on the virus
Mhm.
that you've
Mhm.
introduced, or the
Mhm.
antigens specifically that you've introduced
Okay.
via the vaccine.
Okay, so this makes sense. So what I'm gonna like you had, because again, series 1, our long-term listeners will know this. Find a bat, put it in some Tupperware, send it to Taz, because Taz would get deliveries of Tupperware bats. She would unpack the bats, which she'd go into the lab. She'd have to dress in a full hazmat suit because, as Yaz said, humans can get it, so she's got to protect herself. So she goes into the lab. We're doing like everyday day in the life of Taz. Goes into the lab.
Oh my god, but also let me just say, Taz actually has to like wash her hair and stuff when she's done in the, like, she has to do a full on like cleanse after.
Exactly.
which I
So she
find
goes
mad.
in, shower, like an actual shower, then hazmat suit, go into the lab, takes the bats out the Tupperware. Yaz, you might know this. How does she get the virus out of the bats?
Why would I know that? Molecular biologist. Virology. Let me go back to, I didn't learn virology.
So that was like kind of like a side part of my project, because we had like a database of viruses anyway that I worked with. But like if we got a bat submission, then it could have like a new strain of a particular species of lyssavirus. So we would always check.
So like day to day, you're not getting the bats out of the Tupperware.
My colleagues are, because it's like a routine testing, but it wasn't fully part of my PhD, no.
So we've really made no progress than like what Taz did then.
No, we've basically answered it. She's neutralizing the lyssaviruses that come from bats that have rabies thingies, like rabies viruses.
Okay.
And she's looking at how the antigens are present on the lyssaviruses and how you would like.
Okay. Smashed it. Yeah, we smashed that then.
Is that right?
Perfect.
You kind of got there in the end. Should I give like a rundown of what it actually is, or what?
Yeah,
See
maybe
how close
like
you were?
a quick overview. I think we were bang on. Like, I reckon we could do that
Same.
PhD like so quick.
Same.
Okay, bitch. Took me 4 years, but all right.
Yeah, I could do it in 2.
I did one.
Okay, so the crux of my whole project was I basically wanted to define what you would need to include in a vaccine to be cross-protective against all lyssaviruses. So I think from memory there's like 17 or 18 lyssavirus species. There's a current rabies vaccine that is like a routine vaccine if you want to go to certain countries, and that protects you against 10. So I was looking at whether it does actually protect you against the 10, what level of antibodies that you need circulating in your blood to actually protect you against all those 10. Then I was looking at how we could try and have protection against the other 7. And the viruses can be split up into 3 groups based on how different they are from each other. So you've got phylogroup group 1, which are all very similar and they're afforded protection by the current rabies vaccine. You've got phylogroup group 2, which there's no vaccine protection currently, but they're all really similar and they all kind of originate from Africa. And then you've got phylogroup group 3, which is kind of like just a collection of viruses that are all just really different and really divergent. So I was thinking, like, for a vaccine, you'd probably have to include an antigen of some kind from each of those phylogroup group 3 viruses because they're just so different. And I was also thinking with the phylogroup group 2 viruses, one virus might be representative of all of them. So I wouldn't have to include antigens from all of them for cross protection. So I basically did a lot of virus neutralization assays. So you mentioned ELISAs, and like that is kind of like an assay for looking at antibody binding rather than like antibody neutralization and preventing viruses from going into the cell. So I looked at live virus neutralization assays, which I won't go into depths of like what that actually is, but
I'm not gonna lie, I'm just gonna say quickly, like, Taz is actually like changing the world.
Yeah. Yeah, it's true.
My project's actually, I'm not gonna mention my project.
Not yet.
we're gonna have to in a minute, but
Yeah, literally. Literally.
just to like wrap it up, I basically tried to make like different recombinant proteins to include in a vaccine. Yeah, that was it basically. There's way more to it than that, because there was like new viruses that got detected in the middle of my PhD, and then I made a recombinant virus to include them in my panel and all that sort of stuff. So
Not gonna lie, we always knew Taz was like smart. That sounds so sci-fi film. I'm gonna make a film about you,
Yeah,
Taz.
literally.
I'm gonna
Literally.
direct it. Liv, you can
Yeah.
write it, I'll direct it. I think it would be banging. We can get like
Okay.
someone to look like Taz and dress up in
well,
the,
I
like...
could just play it myself.
We could just have Taz. She can do it all, babes. She'll probably write the whole film and direct it and act in it at the same time.
Teamwork. Okay. Thank you for sharing your PhD topic.
Oh, that was good. That was good.
Very good. Okay, Liv.
I don't actually know mine. Can I Google it? Because
What?
I actually got it.
How do you not know it?
Oh, like I know it roughly, but I don't know.
Yeah,
it.
do the
One
rough.
second. No, like I don't know the title. Like, let me just get.
I
the
can't believe
title.
you don't know that. You worked on it for 4 years. I mean, to be fair, the title is like the thing you just add on at the end.
The title, and also I changed my title like last minute. Okay, my title is actually very broad. So I feel like you're either just gonna like, it's gonna either be really obvious or like it's gonna confuse you because it's so broad. But my title is Influence of External Factors on the Human Epigenome.
Yeah, that's very broad.
Okay, so I do feel like I know stuff that you've mentioned in the podcast previously about like smoking or pollution. Maybe.
Yoga.
I'm nodding. Yoga. I don't know if she did that in her PhD though.
No.
But Taz, you can explain the epigenome.
Do you think I know that? Okay.
Yeah.
Yeah.
All right.
We've done an episode on it.
We actually did do an episode.
This is really testing my knowledge, guys. Right, okay. So I think the epigenome is like the non-coding DNA that
No.
Okay. Well, if the epigenome is basically,
I mean, like, you're on the right
think-
path.
It's DNA.
It's not DNA.
RNA.
What?
No, it's the tags. It's the like markers that go onto DNA, bind to DNA or proteins.
To determine like whether genes are transcribed.
Yeah, kind of. Yeah.
So
You're on the right path.
Is it that they're transcribed? I thought it was just that
Or
it can
expressed.
modify.
Modifies expression of genes.
Mm-hmm. Yep. Taz is right.
But like,
yeah.
surely the epigenetic tags, they bind to non-coding DNA, no? Because they like would bind to like promoters or
Yeah, well, there's like several types of epigenetic tags, if you remember.
yeah. I'm just thinking of the methylation.
Okay, yeah, if we think about just methylation, then yeah, you're right.
Yes.
And then if we've got like
Yes, why
there's
she looks
others.
so confused.
Because I was
Yeah,
gonna say I was gonna name the others, but I've histone markers, and that's all
histone
that I can name.
modifications.
And then the siRNAs.
Well, you've got like non-coding RNAs.
So, what did Liv do for her PhD? She just looked at the whole epigenome. In what? That is actually a very broad topic. You looked at just all external factors.
Smoking. I'm going to say smoking, drinking alcohol. What are other things? Pollution.
So you'd look at like a control person that hasn't done those things, look at their epigenome compared to someone who has like smoked, and then see if their like these tags or markers have changed.
Wow, that's good. I mean, I didn't look at smoking.
Oh.
But
why are we so convinced you did smoking?
I'm sure you did pollution.
Well,
did you do pollution?
I did pollution. Yeah.
Yes.
Oh, and didn't you do sex differences?
Yeah. Wow, you guys really listen to me.
We're basically smart. So, okay, now let's unravel how you did that.
Oh, God.
Let's do the pollution, because that was like my main thing.
So there must be like data banks of, I don't know. I'm not very good at coding. I don't know all this stuff.
So did you have like a specific area you were looking at that was like a polluted and a non-polluted?
No, I was looking at the whole of the UK.
But like, were you looking at like the full human genome?
Epigenome, yeah.
And you were looking at specifically the methylation one?
I looked at a few other
Mostly.
things, but mainly methylation, yeah.
So, question.
Mm-hmm.
How do you obtain the epigenome? And is it that we've got like multiple epigenomes of like people all over the UK that have just been obtained in a database?
Yes, so you can just get it from like taking blood. So you can just take blood from someone, and then you can run like microarrays and measure methylation. So you know like across the genome like where all of these tags are. Obviously that's a very simplified term, but yeah.
Can you do mine?
So, yeah, we could we could take your blood and do it.
I'd just like to know. You could literally tell me so much about myself. Okay, so they've obtained loads of different people. You've got a database, and then you're like comparing between areas, different pollution sites that like are different. No, and like
no.
Okay,
I'm gonna tell you this because I feel like it's gonna be quite hard to get. So around the UK, there's like loads of different measuring sites that like measure air pollution. So basically, what I did was like take all the data from them. Take like everybody whose blood I had. I also had their postcode, so I'd take their postcode, and then basically I made like some geostatistical model to take their postcode, look at how close it was to like the stations, and like I'd use several stations, and then like extrapolate the data to estimate how much air pollution they're exposed to.
Oh, I guess that would be kind of easy if you're like in countryside versus London, because London air pollution is shocking.
Yeah.
But then were you also like doing the sex differences within pollution?
No, that was just like a separate thing. That's why my title was so broad, because I did like several projects within the PhD.
Okay, so you did that like running some code, and then you just got the answer.
Just like that. Just run the code and we got the answer.
That's literally how it seems. Right. Wow. How close were we? What did you actually do?
Like, see, I could do that in 6 months.
Just press enter. Just press run code. See ya. And then like all your plots just like sitting at your computer like enter.
That'd be so good if you could automate that well.
If what was it? No, a for loop. Just run that stuff. If there's pollution, if they're like 20 miles away from the site, don't include their data because they're too far. Oh, but distant. I don't know. Okay, that could be a lot. That's like a lot of factors. Did you run a lot of stats on your data? Was that just like
loads of
ANOVAs? Oh, I only know an ANOVA.
No, look, I'll break it down for you. Really simple, right? We got the blood, we got the data with like where all the tags are in the DNA. Then I also know like all of these different pollutants, how much you've been exposed to, because I've got your postcode. And then what I do is I run a linear model to see basically if there's like a correlation between high pollution and this tag at this specific site in your DNA. Yeah.
I would
So
have
then I
said that
know,
linear model.
and then I just do loads of stuff to find out why that tag might be there, what that tag might do, what it might mean for disease, blah 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 blah. Obviously that's a highly simplified version, but I'd say you guys were pretty close and I'm pretty impressed.
I think that's
That's
good.
so
But-
interesting.
One question.
Yeah.
When you looked at how that methylation on a certain site could be impacted like with disease, was that more like literature searches? Because you weren't then in the lab doing anything. Like, how do you correlate the things that you found?
So I'd look at like genome data to see, like, for example, what gene it's in and then where it is in the gene, because like where you find methylation in a gene impacts like whether or not it actually influences gene expression. So like, for example, if it's in the promoter of a gene, then it's more likely to switch the gene off. And so I'd look at that. Then I might also look at like databases of like histone modifications to see how they interact with that. I also did a lot of like chromatin looping stuff, which I've explained before, like, you know how your DNA is like super long, but obviously it all has to like fold up to fit inside your cell. So I did a lot of work where I would like calculate how it folds up inside your cell, and then like how
I've got a question. So you obviously you worked out like how the DNA folded up and stuff. Did you find that, like, I'm assuming like with the way that DNA folds, you could have like one part of the DNA that's like linearly is so far away from another point, but when they fold, they like are really close together. Did you find that like if one point was methylated, like that point that's like folded really close was also methylated, but like linearly it would be hard to like make that relationship because they're like so far away from each other?
Taz, you're so clever. That is exactly it, because like, for example, you might find a tag in a gene that has like no relevance to the disease. So right now I'm looking at like Alzheimer's, for example. So we might find a tag in like a gene that has nothing to do with Alzheimer's. It's never been reported to have anything to do with Alzheimer's. But then if you look at, for example, at like chromatin looping and calculate like how the DNA loops and folds on itself, you might find that it actually does then come into contact with a known Alzheimer's gene, and like that's why the tag exists. And then you then have like a new mechanism for like how epigenetics might be influencing like Alzheimer's. So
Sick.
You lot are also smart.
I mean, we're all smart. We've done
Yeah.
PhDs.
Yeah.
True.
I always
It just
find it so
sounds like
interesting to learn about other people's research, because then I'm like,
Same.
oh, I want to do that.
Yes, same. I'll do it in 6 months though.
But yeah, like basically to summarize, in answer to your question, yeah, it's like we, like people like you guys, you do stuff in the lab and then you make your data like publicly available. So in order to calculate the chromatin looping stuff, someone had to produce high C data in the lab, but then they make it available, and then I like go and pick up the data, and then I do all of the like statistical stuff with it. So
Where do you find those data banks and stuff?
like Geo is like the biggest one. Gene Expression Omnibus, I think that's what it's called, Geo.
Oh, okay.
But there's like loads. Like if you go into any like paper, most often people make their data available, right?
Okay,
Like
yeah,
their raw data from the lab. They should, because then we can do extra stuff with it. So like, yeah.
Damn.
Cool.
Look at us.
So well done, guys. I'm proud of you. But yeah, I did like pollution. I also did sex differences, and then like a bunch of other stuff. And I did kind of look at like smoking and stuff, but it was more just like including it rather than focusing on it alone. But yeah,
Love that.
let's do you, Yaz.
So my title is Novel Tools to Unravel the Phosphate Calcium Signaling Pathway in the Marine Diatom Phaedactylum tricornutum.
I hate that freaking organism. I'm sorry.
Backstory: I did a side quest, a project, like a research assistant project on this diatom, and we're so behind in the field. Like we have the genome sequence, but there's just like nothing, like no help available. So
it's tough. It's tough working in that field. Working on that organism was tough. Like, don't recommend to anyone. So I don't know how you do it, Yaz.
Yeah,
because there's literally no data. Like nothing is freaking known about it.
But then
so that's where the
find
novel
is, like,
tools. Wait, say it again. Novel tools.
Yeah, what is it?
Novel tools to unravel phosphate calcium signaling in the marine diatom Phaedactylum tricornutum.
That's bare easy. Basically, you're just trying to figure out the phosphate calcium signaling pathway.
Exactly.
And you're just got to be the one to figure it out.
Exactly.
1 month. Could do that in 1 month. Easy.
As we've gone through the project, it was like, okay, 2 years, 1 year, 6 months, 1 month, bro. Come and join the lab for 1 month internship or something and get it done. Just do it.
Okay, so Phaeodactylum is it an algae?
Yeah, it's a diatom, which is the type of algae.
The actual audacity of me, like I've done a full project on this and I'm like, what is it? So you get your algae. Where do you get your algae from? That's the question I have.
Like, it's been in culture for like 100 years. So I'm not a boat-going, sea-going marine biologist. I just go to my culture room. So it's like an 18-degree incubator room. And then we have different lights. So our algae grow in a different light schedule. And then we just grow them in culture flasks.
Okay, so you go to your fridge, culture room, get your algae, put them on the plates, do your little like thing where you get you know what I'm talking about? What's that called?
Streaking the algae on.
You streak your algae.
Yeah, on an agar plate.
And then.
Oh, that's cute. It's like bacteria.
Yeah, that's why I'm a microbiologist.
Am I actually right in saying that? Because I thought you were going to laugh at me. You actually streak it.
I do streak it, but it depends. I streak it like a culture. Yeah.
Oh my God, yeah. Cute.
But what is the phosphate calcium signaling pathway? Like, what am I actually trying to understand?
Good question.
Phosphate calcium signaling pathway. Actually, no idea what that is.
Think, like, make it up. Obviously, I use cultures in the lab, but where do diatoms normally live?
In the sea.
In the sea.
What would you say, live?
Like, think about like on the rocks of the sea, because that's where algae is. But like in the sea.
Yeah, in the sea.
In the sea. So the phosphate calcium signaling pathway helps them to, like, what is the purpose of the phosphate calcium signaling pathway?
Live in the salt.
Ooh,
because the sea's like sodium chloride, Na. I don't know.
But we're talking about phosphate calcium, babe, not sodium chloride.
Yeah, I was getting mixed up with calcium and chloride.
Calcium and chloride.
Okay, the phosphate calcium signaling pathway, I think, helps them to exchange nutrients.
Yeah.
Is that it?
Yeah.
Oh my God, check me out.
But Taz, you're also on the right track.
What did you say, Taz?
Oh, I said phosphates are used a lot with like activating and deactivating enzymes in pathways.
Mm-hmm.
So like if something's phosphorylated,
oh, so it's like, is it like how they eat and stuff, like how they get their energy, or like like their equivalent
oh my
of eating?
God,
you know
no way. What is it there?
Is
like
that sarcasm?
equivalent of like what's it called? Photosynthesis.
So they do
Yes.
photosynthesize anyway, because they are photosynthetic algae. So they have that pathway, so they can produce oxygen, take up carbon dioxide from the atmosphere.
I've got it. You're
go.
trying to understand how that works. You're trying to understand that whole process, because
What process?
Phaeodactylum, the like how they like exchange like their version of photosynthesis. You're trying to understand that whole process in
So it's not-
Phaeodactylum.
Their version, they do photosynthesis separately. Phosphate's not got anything to do with that necessarily.
It's like another additional energy source of some kind.
Yeah.
So like how they interact with the environment, I guess.
Yeah.
And like, so you're trying to understand that, because you told us before that algae, and I'm guessing Phaeodactylum, are like going to be really useful for like bio stuff, and you're trying to understand it so that like we can use algae as more sustainable and alternative like resources.
Yeah, so that's definitely one part of it. My project isn't so biotech focused, but that is a very like big part of it. But algae or diatoms can also be bloom forming. So in the ocean, they can like cause these big algal blooms, which can cause problems to like fish stocks. They can cause toxicity to some species. And then like if we ate mussels, you could get shellfish toxicity from them, because some of the species of like phytoplankton could be toxic. And we just don't know anything about how they respond in the environment. So what we're looking at is how phosphate, which actually every organism needs as like an energy source, because what Taz was saying is like phosphate's used in like RNA, DNA, protein, like post-translational modifications, if you've got a phosphate group in addition. They're also used for like stability in the cells. So you really need phosphate, but diatoms don't have a brain. So it's like, how do they interact with their environment when it's readily available or when it's not? And because diatoms just like float around in the ocean, they don't have control of like, oh, we need to go to this high nutrient supply. They just like have a real big change constantly. So it's like, how are they able to like manage and maintain different fluctuations of phosphate in the ocean? And so I'm looking at the phosphate calcium signaling pathway. So now I've said that, I want you to try and think of like, what is this calcium signaling pathway? Like, why would you use calcium in a pathway? And I'm thinking it's more like biochemically relevant.
Isn't it like something to do with like sodium, calcium channels, or no?
Yeah.
Like it opens some channels to allow
Yeah.
things into the cells.
Yeah. That is literally it. So calcium
I'm
can come
so
into-
clever.
You're actually
My
so clever.
God. So I'm thinking like you put these algae on the plate, you streak them, and then you then expose them to like different levels of like nutrients or different levels of starch, and then measure like maybe the calcium channels, and like that's how you understand like how they respond to different exposures, I guess.
Yeah.
Oh my God, yeah. And didn't you link it to like calcium and a fluorophore or something? And then you would kind of measure if they're taking up calcium or something, and then they would like fluoresce under your that's your
oh my God, yes.
We smashed that.
You
We
actually did. So,
smashed that.
We have a fluorophore, so like a fluorescent tag. So we genetically transformed our model diatom to show a change in cytosolic calcium, so within the cell. So when we have our diatoms grown in really like limited or low levels of phosphate, but then there's suddenly like an influx of phosphate, we see that calcium elevations within the cell increase. So this basically means that because algae don't have a brain, they've got a different way of like having a nervous system. So what they use is calcium, and the calcium then comes into the cell being like, hey, we've got phosphate available, and then triggers downstream proteins to like function or causes like
that's
gene
kind of
expression
cute.
regulation. So then I'm looking at like all the mechanisms of that, so including like calcium ion channels, looking at like calcium binding proteins that would cause downstream phosphorylation, and then I'm also looking at how the phosphoproteome changes within like calcium signal.
Damn.
So yeah, you
We're
guys
all
got there in the end.
clever. We are actually
Wow,
all
look
clever.
at us.
Yeah,
I'm actually really impressed with all of us. Like how we actually
Yeah,
just
me
like
too.
we just dissected those titles like it was nothing.
nothing.
We could
1
do that
month
all
done.
day. We could do that
everyone
all day.
send in your PhD title and we'll
Oh my
do
God,
like
yeah, we
a
should
mini
make it a
series.
thing.
That
Then
is
we
a
wouldn't
good
have
idea.
them.
LaboraTEA next season. Our LaboraTEA is your PhD titles unraveled.
My God. That is a good idea. So we did the what would you do, Ebs? We did the polls. I really want to dissect some of the listeners' titles, but what we're going to need you to do is send in your titles and then also like a 4 or 5 sentence explanation, just so we can know if we got it right. This is perfect. This is what we're doing next season.
Amazing. I'm proud of us. We're actually smart.
We actually are, but I mean, I guess that goes to show like how transferable like all of science is. Like we're all in completely different fields, but like general terms you can break them down, and like I'm proud of us.
And I think this is such a nice contrast from last season. I don't retain information like for a quiz. However, if I need to interpret something using my knowledge, I've got you guys. I can get there eventually. So winning. We are winning.
Proud of us.
Proud of us.
Okay, well that actually wraps up the whole of season 3. I can't believe like season 3 is done already. And do you know what? I'm actually genuinely, like I said this last season, but like this season I'm actually really sad to see go, because I know that so many people have absolutely loved this season. We've got so many non-scientists actually who have been listening to this season who have really enjoyed it, because I feel like films have just been something that's been so relatable to like everyone, whether you're a scientist or not. So I feel like there's one thing that I've learned from this season is that like something we should do as a podcast is really try and make all of our seasons and everything that we talk about relatable to everyone. Do you know what I mean?
Yeah. Obviously, this is the last episode of season 3, and like Liv said, we are so sad to see it go, but we will be back for season 4, and we already know the theme for season 4, which we're super excited about. So make sure you're following us on Instagram at So Cultured Podcast, where we'll be like revealing that in a few weeks' time. So yeah, just keep up to date with that. So yeah, I guess that wraps up season 3, guys.
Can't believe it. That's gone so quick. Season 4 is gonna be mad because the theme is even better than this season. But we say that every single season. So I don't know.
Yes,
Like,
do you actually think it's better? I feel like this season is always going to hold like a really special place in my heart. But what I will say about season 4 is that, again, I feel like we've kept it like it's going to be something that's really relatable. But actually, something we wanted to do with season 4 was give you guys science that you can actually implement and action. So that's like a hint for what season 4 is going to be. But I don't know. Yeah, what do you prefer, season 4 or season 3?
Liv, you might be team season 3, but I promise you, season 4 is where it's at. Like, I'm hyping up team season 4.
Okay,
and Taz,
I believe you.
You're not allowed to be on Liv's team this time. Like, you gotta just be with me. Season 4 is where it's at.
Yeah, season 4 is where it's at. Or ask me at the end of season 4 and I'll let you know.
Okay, so remember, guys, we will be back. Stay up to date with us on our socials in the meantime. But just a really big thank you to everyone who's tuned into this season. We know we've got so many new listeners, and we just love and appreciate you guys so much. And we will see you back for season 4.
Bye.
Bye, everyone.
Bye.
See you soon. You've been listening to the So Cultured podcast. Thanks so much for tuning in.
Make sure you listen in next week for more science and tech updates and the hottest LaboraTEA.
You can stay up to date with us in between episodes by following us on Instagram and X at So Cultured Podcast.
See you back here, same time next week.
Just watch me